Your brain, it's this incredible buzzing electrical storm, right? Just constantly firing signals that let you think and move and feel. But what keeps that whole system from just spiraling into total chaos? Well, today, we're talking about the brain's absolutely essential off switches.
I mean, just try to wrap your head around that number. 86 billion neurons, and every single one of them can fire off signals hundreds of times a second. That is a staggering amount of on signaling. So what puts on the brakes? The answer is a crucial but maybe underappreciated part of the story: inhibition.
You see, the brain is really just a constant, delicate balancing act. On one side, you have these excitatory signals, mostly driven by a neurotransmitter called glutamate, and it's basically screaming, go. But on the other side, just as important, you have inhibitory signals, mainly from GABA, that are whispering, stop. Without that stop signal, the whole system would just short circuit.
So here's our game plan. We're going to start with that balancing act, then we'll meet the brain's main brake system. We'll see how it works and how some pretty common substances can hijack it. Then we'll meet its lesser-known counterpart and wrap it all up with a really fascinating plot twist.
You know, a healthy brain isn't just about firing neurons all willy-nilly. It's about firing the right neurons at exactly the right time. And inhibition is what gives the brain that precision. It carves out the signal from the noise and prevents overexcitation. It's truly the silence between the notes that makes the music.
All right, let's meet the star of our show, GABA. It stands for Gamma-Aminobutyric acid, but nobody calls it that. This little molecule is, without a doubt, the most important inhibitory neurotransmitter in your brain. In fact, get this, it's estimated that maybe up to a third of all the synapses in your entire brain use GABA to just say, hey, calm down.
And here's where it gets kind of ironic. The way the brain builds this brake is pretty clever. It starts with glucose, sugar, and metabolizes it into glutamate. And remember, that's the main go signal. Then this special enzyme called GAD swoops in, and with one simple chemical tweak, it flips the accelerator molecule into the brain's primary brake.
Now that GAD enzyme is the real MVP of this whole operation, but it can't work alone. It needs a crucial little helper, a cofactor that comes from vitamin B6. Now that might sound like a tiny biochemical detail, but its importance in the real world is, well, it's staggering.
Back in the 1950s, a terrible accident happened. A batch of infant formula was made without any vitamin B6, and the result was a tragedy that perfectly illustrates this chemistry. Without B6, the GAD enzyme couldn't make GABA, so the brain lost its brakes. That led to uncontrolled electrical storms, seizures, that were, tragically, sometimes fatal. It's a harsh lesson in just how vital this system really is.
Okay, so GABA gets released into the synapse. How does the next neuron actually get that message to slow down? Well, it all comes down to the receptors. Think of them as molecular locks that GABA acts as the key for. And there are two main types, each with its own style of getting the message across.
So neurons are listening for GABA in two main ways. On the left, we have the GABA-A receptors. Think of these like a simple light switch. They're direct, super fast acting. GABA binds, a gate opens, and the message gets delivered instantly. On the right, you have the GABA-B receptors. These are a bit slower, more indirect. They use a kind of internal messenger system to deliver their stop signal.
Let's really zoom in on that GABA receptor, because it's the brain's most important inhibitory gate. It's a pentamer, which just means it's built from 5 protein parts that form a little channel or a gate through the cell's wall. When GABA locks in, that gate swings open, and a flood of negatively charged chloride ions rushes into the neuron. All that negative charge makes the neuron less likely to fire. It's kind of like pouring cold water on a spark.
Now, because this system is so fundamental to calming the brain down, it's a prime target for a whole bunch of drugs and other substances. A lot of the things we might use for anxiety, or for sleep, or even for recreation, they work by hijacking these very same GABA brakes. For instance, take a look at this spot right here on the receptor. This is where benzodiazepines, drugs like Valium, bind. What's so cool is that they don't actually open the gate themselves. Instead, they're more like a master key that just makes the lock way easier for the natural GABA key to turn. The result? A boosted stop signal, which is what gives them that calming anti-anxiety effect.
And it's not just benzos. Barbiturates and sleeping aids like Ambien, they target this GABA-A receptor too, just usually at slightly different spots. They also crank up GABA's effect, but their action can be much stronger, leading to heavy sedation or sleep. It's just amazing that this one tiny molecular machine has so many different knobs and dials that can be tweaked.
And yep, you guessed it, even alcohol gets in on the act. Ethanol targets the part of the receptor that's actually tucked into the cell membrane. And by boosting the GABA receptor's function, it helps cause that loss of coordination, the slurred speech, all the classic effects we associate with being drunk. It's really all about pushing harder and harder on the brain's natural brake pedal. So, GABA is the undisputed king of inhibition in the brain, no question. But it's not the only player in town. If we travel down the nervous system to the spinal cord, a whole other molecule takes center stage.
And that molecule is glycine. It's the simplest amino acid there is. And while GABA is running the show upstairs in the brain, glycine is the primary brake for the spinal cord. It's in charge of managing all those motor and sensory signals, and it's used in about half of all the inhibitory synapses down there.
And check this out. Its receptor works in the exact same way as GABA-A. It's another chloride gate. Structurally, they're practically cousins. And here's a little memorable and kind of chilling fact for you. This receptor is the specific target of the poison strychnine. Strychnine blocks these gates, it removes the brakes on the spinal cord, and that leads to horrific convulsions and muscle spasms.
Okay, so we've established that GABA is the brain's ultimate stop signal. It's the brake. But as you've probably figured out by now, the brain is rarely that simple. So prepare yourself for one last fantastic plot twist. What if I told you that in the developing brain, GABA is actually an excitatory neurotransmitter? I know, it sounds crazy. But look, it all comes down to the concentration of chloride ions inside the neuron. In immature baby neurons, chloride levels are really high. So when GABA opens that gate, the negative ions actually flow out, not in. And that makes the cell more positive. It excites it. It's only later, as the brain matures, that a special pump kicks in, lowers that internal chloride, and allows GABA to become the inhibitor we know and love. Yeah, you heard that right. The very molecule that will spend its entire life calming down neural circuits actually starts out by revving them up. It's a complete and total role reversal.
And this isn't just some weird developmental quirk, it's absolutely essential. That early excitatory kick from GABA is thought to be crucial for basic things like telling neurons where to go, how to grow, and which connections they should make. It literally helps build the house that it will later be in charge of keeping quiet.
It's just a stunning example of the brain's complexity and elegance. The very molecule that's tasked with providing the essential stop signal to keep those 86 billion neurons in balance, it has to earn its job. It starts its career doing the exact opposite, proving that in the brain, even the brakes have to begin their life as an accelerator.